Good morning, everybody. My name is Paul Hargreaves. I work at SRUC, and I'm welcoming you today to the AHDB VESS Agronomy Roadshow. And today we're going to be looking at soils and soils quality and the health of the soils and how to maintain that. So I'm hoping it's going to be an interesting morning. We've got 3 presentations that are going to be I'll be introducing, and then there'll be a chance for some questions and hopefully some answers after that. So we go for the first presentation, which is made by Joanna Cloy, who is a soil scientist and has been working as a researcher at SRUC, Scotland Rural College, since 2011. She's completed a PhD in environmental geochemistry at Edinburgh University, and Joanna's current research involves investigation of agricultural systems under different management activities, assessing soil greenhouse gas emissions and mitigation, soil structure and soil quality, carbon storage and stability, and nutrient dynamics. And she also delivers postgraduate and undergraduate teaching at Edinburgh University and for the agricultural consultancy with SRUC Consulting. And her presentation really is going to be an introduction to soil and be looking at the chemistry and the nutrient cycling within the soil. So we'll hear from Joanna now and her presentation.
Hello, I am Dr. Joanna Cloy, and I am pre-recording a talk on soil health for the AHDB Roadshow. I will now share my presentation slides. Okay, so I'm going to give an introduction to soil health and how it can be measured in terms of chemical and physical properties. So this is a brief overview before Paul and Christine give their talks that will build on what I've just spoken about. So soil health has appeared in news headlines quite a lot recently, usually quite bleak news about how soils are being degraded. There is 3 times more carbon held in soil than in the atmosphere, but carbon has been lost due to intensive farming and deforestation. And these activities fuel climate change and compromise attempts to feed the world. So, of course, we need to take action now. And with Trump no longer in power, and with ministers in the UK accepting that farmers need incentives to farm in a way that leaves a soil healthy for future generations, perhaps we need to think positively towards the future and how soil protection is a core issue of improving our soils in the future through the agriculture bill, for example. So what is a healthy soil? Well, farmers and growers have taken the initiative to understand the health of their own soils and use on-farm approaches to monitor and optimize soil health. And our research really tries to help provide the evidence to promote good practice so that we maintain healthy soils. But we need a set of easily understood and measured indicators. Of course, we can tell if a soil is healthy through how it looks, feels, smells, but there's a lot of other simple measures that can be done. So let's just have a look at what soil health is. Apologies if you have soil health fatigue, just like COVID fatigue, but it is a crucial way of defining the function of our soils. Physical, biological and chemical components together need to be considered, and we need certain approaches that build on the traditional kind of chemical approaches that have been done typically since historic times. We need to start thinking about the physics and the biology in conjunction to just your traditional soil nutrition and pH type standard measurements in the lab. And of course, there's a lot of exciting research that's going on with trying to find the evidence and the best indicators for soil health. This table lists some of the key chemical indicators of soil health. Things like pH and nutrients, like I've already mentioned, but then there's also things like cation exchange capacity, carbon to nitrogen ratios, electrical conductivity. Loss on ignition, that's the same as organic matter, soil carbon, total nitrogen, hot water extractable carbon, that's a way of extracting the labile, high quality carbon in soils, the carbon that feeds the biology. And similarly, there's the light fraction of organic matter, the less decomposed material that's more edible to the organisms. And then potentially mineralizable N as well. So I'll talk about some of these in the talk. So of course, the most important, soil pH and nutrient status. It's probably the most important properties for defining soil health, and the standard soil test system for nutrient testing is pH centered, and it's based on the concept of maintaining sufficient nutrient levels, and it's simple and low cost. We know that if the pH isn't right, nutrients will not be available. So soil testing is critical. Testing for liming requirements, perhaps typically every 3 to 5 years on fields that have the most economic value, let's say. And SAC consultants have produced these technical notes that give guidance for pH values for different systems. And also there's many technical notes providing guidance on, specific trace elements or how best to manage phosphorus. So it's critical that the advice that we give is linked with research and evidence. And the photographs here just show how grass and clover is really struggling at pH 5.5 and lower. So we have long-term experiments that provide the evidence for the impacts of pH on soils and yield. Again, with this one, you can see that spring barley clearly suffers at the lower pH ranges. So nutrient budgeting, as well as pH testing, is of course also important, as you don't want to apply too much nutrient. You want to work out what the crop offtake was and try and make sure that your soils are on target. Make use of any residual nutrients in the soil and apply fertilizer accordingly. So of course timing is critical. Timing isn't always the easiest with the unpredictable weather, but using a proper nutrient budget will not only help business, but also the environment, reducing diffuse pollution. Another issue is using the right liming product. So if you have high magnesium in your soils, they'll be tighter, stickier soils. So it's best to use the calcium lime rather than mag lime in that case. And some researchers believe that it's this kind of ideal ratio that should be aimed for. And soil testing can help check whether you do suffer from an excess of magnesium. Let's move on to soil organic matter. So one of the properties that we can measure for soil health in terms of, well, chemistry and biology, but also physics, as it kind of underpins a lot of the functions in terms of chemistry, physics and biology. And soil organic matter has been described as a major currency in the soil system. So we know that to increase organic matter accumulation in soils, we need to reduce the losses through perhaps intensive cultivation or not returning organic material back into the soil. And soil organic matter is typically ranges from 5 to 10% in Scottish soils, and organic matter is comprised of around 58% organic carbon. So when we're talking about organic matter, we're talking about organic carbon, and we can't escape talk of carbon at the moment with climate change. Potentially mineralizable nitrogen. This is another measure which perhaps needs more research for us to fully understand how we can tap into this kind of mineralizable form. Of course, it's organic nitrogen that needs to be mineralized through biological processes to make it available for plants. And we do, in the lab, we do incubations where the potentially mineralizable nitrogen is determined by basically incubating some soil and optimizing factors required for soil microbial activity in one set of soil and compare it to a control. And then it gives a measure for how available that nitrogen can become once incubated under the optimal conditions. So it's basically a way of defining the capacity of soil to supply N for crop growth and helping eliminate overuse of chemical fertilizers. This table indicates some of the physical indicators of soil health. So, of course, water content, water retention properties, available water capacity, bulk density gives a measure of compaction, particle density, stoniness, visual evaluation of soil structure, the permeability, porosity or water-filled pore space. So, of course, soils consist of air, water and the solid matrix, the mineral and organic matter. And of course, if most of the pores are filled with water and there isn't a kind of favourable mixture of air and water filled spaces, then we know that the soils are not going to be in a healthy condition physically. Infiltration rates are quite easy to measure. And then we have other measures that require maybe perhaps more detailed investigations in the field or lab, such as the rate of erosion and aggregate stability. Soil texture we can do through hand texturing quite simply, and a simple device such as a penetrometer can be used to detect compaction layers. Cone resistance or penetrometer resistance and shear strength are just measures of soil strength. So let's have a look at some examples. So here's the VESS scoring sheet. VESS, the visual evaluation of soil structure, is a simple spade test for visually assessing your soils. All you need to do is dig out a spade-sized block of soil, break up the block by opening it up like a book, and then kind of looking at these indicators based on the photographic keys. Look at whether the soil fits into the green, amber or red category based on appearance, also based on how easy it is to break up those clods or clumps of soil. If there's not many big clods or big compact layers, then it's a sign that you've got healthy soil quality, soil structure. And porosity is another issue. Having a mixture of pore sizes is a good sign for soil structure. If you've got distinct macropores, like wormholes, where the roots and the worms have to really force their way through the soil, that's an indicator that you've got compaction, you've got a compact layer. And of course, if you have a grey-blue colour as well, it will indicate waterlogging and compaction. So ideally, you want to have the soils in that top row of the chart here, where they're friable, they smell good, they're easy to break up, and the roots are going throughout. What do the scores mean? So scores of 1 or 2, of course, are good. Also, if you have a 3, it's a fair score, but the 4 or 5 really will indicate that you've got poor soils and you maybe need to think about trying to alleviate the compaction or alleviate the waterlogging problem. So soil strength, as I said, can be measured using a penetrometer, such as the one shown on this slide here, and it measures the resistance of soil to root penetration. So you can imagine that it mimics how easily roots can navigate through the soil. And soil strength depends on compaction, but of course it depends on texture of the soil, and some soils are naturally compact due to their texture. Stones can be a pain, and also if the soils are too dry or too wet, then it's a kind of difficult measure to use. But if you do it at field capacity 1 or 2 days after rainfall, then you should get good results, and it helps kind of test against the visual evaluation of soil structure to see where the depth of compaction is occurring, so that you can carry out your sward lifting or your subsoiling as appropriate at the appropriate depth. And the figure here is just showing how cone resistance was found to be much better in rows and between rows compared to tram lines, as you would expect. The vein shear strength is another way of measuring soil strength, and it's a term used in soil mechanics to describe the magnitude of shear strength that a soil can sustain. I mean, these instruments are quite expensive, so it's not something that can be easily used by farmers and land managers, but it's a good validation for the simple techniques such as the visual evaluation of soil structure or the penetrometer, which can be purchased at around £300, so it's more of an accessible kind of technique and indicator. Another indicator, physical indicator, is water infiltration. And we use these double ring infiltrometers to help prevent water moving horizontally instead of vertically, because of course we're interested in how quickly the water can penetrate through the profile. But you can use bits of drain pipe and a ruler and a timer, and you can try and determine water infiltration rates for your soils, as it's quite an easy, simple technique if you have the time. So let's think about the measures that can be taken to improve soil health, well, this diagram shows some of the kind of key management practices for improving soil health in terms of the chemical, physical and biological properties. Key guidance is to monitor changes in soil health if you're really serious about trying to maintain your soils in a good condition. Do regular checks, just like you would your car. If you've taken it for an MOT or a service, you want to know what kind of condition it's in. Build up a long-term picture using photographs. There's even apps, VESS apps, so that you don't even have to try and score your soils yourself. You can get the technology to do it for you. There's no apps for counting earthworms, but earthworms are a great indicator of soil health. So simple measures can be taken. Of course, there's no one size fits all. All soils are different in terms of their textures and natural drainage properties, situation, aspect, and so on, climate. So we have ongoing research and developments where we're trying to improve the biological indicators, the research behind them, and also we're gathering data to try and benchmark soil health across Scotland. So the future can be very positive if everyone makes a change to improve soil health in the UK. And SAC Consulting, with assistance from researchers like myself, have developed a soil health test. So it's like an MOT for your soil. Those indicators that I've talked about have been reduced to those listed here, which we decided were the best indicators of physical, chemical and biological health, and they're also the simplest to use. So informed decision making in relation to soil management can be supported through tests like this. I just want to acknowledge colleagues and funding support for the work that we're doing and for this presentation. And thank you very much for listening.
Oh, well, thank you very much for that presentation, Joanna. She can't unfortunately be here with us to answer questions about that later, but myself and Christine Watson will be doing that after all three of the presentations. The next presentation is by myself. My name's Paul Hargreaves. I work at SRUC and usually under normal conditions is based down at in the southwest of Scotland at the Dairy Research Centre, where I've been investigating the soil's effects on forage, especially grassland. But today my presentation is looking more at the soil biology, which is something that's coming more to the fore as we get more understanding of the soil biology. And so I'm going to have, during my presentation, I'm going to be looking at aspects of measuring this, some of the more modern techniques and the more cutting edge methodologies, but also some of the ways that you can measure soil health, hopefully in your own soils as well, and how this potentially will tie in with what Joanna's just been talking about on the soil physical aspects of the soil and also the soil chemistry. So if we go to that presentation, thank you. Hello, thank you for inviting me to speak today. My name is Paul Hargreaves, and today I'm going to be talking about the soil biology aspects of soil quality, how important this is, how more important it's becoming over recent years, and what indicators we could use to indicate soil quality through the biology of the soil. Soil biology is one of the three components of soil health. You may have already heard a little about the importance of the soil chemistry and the soil physics to soil health, and these are relatively well known, and quite a bit of work have been done in these areas, especially on soil, the chemistry of the soil and what nutrients you need to add for good crop growth. However, soil biology has become more important over the recent years, and more investigations and methods for investigating the soil biology have become available. So the importance of the biology of the soil is now better understood. So how much biology is there in the soil? If we think of how many sheep equivalent of soil biology is below ground, how many would we expect that to be? Would it be one, or more likely much more than that? In fact, it's about 100 sheep's worth, or 5 tons per hectare of biology within the soil itself. So we can just think of soil as a living quantity. It's got this organisms living within it. They interact with each other and with the plants that are grown or growing within the soil itself. And with fauna, we're looking at about 1 to 5 tons per hectare. With fungi, another 3 to 1.5 tons per hectare, and bacteria, another 1.5 tons per hectare. So there's quite a great deal of activity going on within the soil itself. And we're understanding more and more now about the diversity of the organisms within the soil. Until recently, these have been really investigated through plating them out on media and seeing what grows, but this can also be related to the type of media that the organisms are provided with to grow, and may not include all the organisms that are contained within the soil. So recently, more sophisticated techniques have been employed using DNA extractions from the soil to look at the number of genotypes of, say, soil bacteria. And these are coming up with very large numbers of different species within the soil itself. And this but diversity of organisms within the soil and the soil biology is being related to what's growing within the soil itself. And we've seen that there are relationships between the diversity of plants grown within the soil and above the surface to what's going on with the below ground diversity. And these also cause interactions. They can cause more help with more resilience within the soil systems, with more organisms there. They can adapt and function in different conditions, and so help to maintain the resilience of crops within the soil itself. So there's more information and more research being done on this recently, providing more fascinating insights into what's going on. We saw biodiversity. For a long time, it was just classed as one black box as soil biomass, the living components of the soil. But with these newer techniques, have been able to be broken down, and we can look at the structure of these and the amount and the numbers of the different components within the soil biomass in a bit more detail. And we're finding that with bacteria, fungi and algae, we're looking at tens of thousands of species within the soil. With protozoa and nematodes, more advanced organisms, there's probably hundreds of those within the soil. And then with insects, mollusks, worms, we're looking at hundreds of these within the soil. Mammals, quite a few, but there's also the plant roots that are important, with although limited in number, are important in the makeup and the diversity of the organisms that we find within the soil. And all this leads to quite a complex situation, with a kind of food web that builds up through the bacteria, through the fungi, up to the nematodes and earthworms and other arthropods that are then fed on by birds and other animals within the larger biodiversity, the ecosystem around what's being grown and what lives within the soil. So these are important as they move up these kind of food web structures. So what's going on in the soil does have effects on bird populations and animal populations as well. And these are some interesting photographs using inflorescence to highlight the living components of the soil. And here's a bacterial colony that's found within the gaps between the soil particles. And you can see there's large numbers of these spread out in these areas where there's food is available. They help to turn over the nutrients that are added to the soil to make those available for the plants that are growing in the soil. And with crop systems, this is the nutrients that are made available to the growing crop. So these are important for good crop yield, good crop growth. There's also the fungal growth that you can see. These are, again, inflorescent photos of fungal hyphae, and these are very important within the soil as they help to hold soil particles together and make the structure of the soil particles as they are found within the soil themselves. So for a good tilt of the soil, then fungal hyphae are very important. These are disrupted and it breaks down the soil particles more. And then we come on to more advanced creatures or more developed creatures, such as the nematodes, which is a small worm-like creature, usually seen with a microscope. And these have got their own ecosystems themselves. They've got predators and herbivores within the different communities, and they can feed on each other, but also they feed on the dead material or the crops themselves. And some of these can be quite detrimental to the crops themselves, causing potato cyst problems within the potato crop. So they're not always beneficial, and it's important to know what you've got there and how to control these. Earthworms are something that are visible, that we can see these when we dig up the soil, so they're potentially a good indicator for the health of the soil. They don't move very far, so they're an indicator of what's happening in that particular area of the soil. They can live for a number of years, so they can see how changes in the soil are affecting earthworm numbers. And they're exposed to the soil itself, so any changes within the soil can affect the earthworm numbers, so potentially a good indicator of the health of the soil. But there are other biological indicators of soil health. So you can use laminar bait sticks, which are a measure of biological activity within the soil. These are relatively small sticks or rods that are pushed into the soil, so basically more interested in the top part of the soil. But this is important as far as crops are growth is concerned, because this is where a lot of the nutrients for the crop growth is found there. You can look at the respiration, so how much CO2 is produced and how much activity is going on within the soil. Again, as I said before, you can consider earthworms and their activity. There's different assays to look at the potential activity of the turnover of some of the nutrients that you find within the soils can be an indicator of soil health. And again, as we said earlier on, the new DNA techniques for measuring the abundance of species are also important as a matter of soil health. So look at the diversity and how these different groups are interacting with each other. There's a microbial biomass where you can just look at the total amount of microbes within the soil and see if that changes with different soil activities or different management of the soil. And then there's looking at various microarthropods or insects within the soil to see if that is any indication of changes in the soil structure. And then the microbial community structure, so using the DNA again to look at the variety of different bacterial and fungal communities within the soil. You can look around the root itself, which is a major area for activity within the soil, because roots can tend to be quite leaky. There's lots of nutrients come out of there, but also some of the soil biology is positive as far as root growth is concerned and can work in conjunction with the roots themselves. And there's the nematodes we've already mentioned. Again, they've got their own community structure. Some can be positive, some can be negative. So it's important to understand what types of nematodes you have got within the soil. And then you can look at just the total fungi and bacteria within the soil. But however, earthworms are some of the more visible components, living components of the soil, and they're not just as simple as the earthworms in the ground. There tends to be three main types based on where they're found within the soil. So there's those that are very shallow burrows that live on the surface of the soil. They utilize the litter layer of the dead material that's left on the surface of the soil and bring this back into the soil. There's one that are more shallow burrowers, that down to about 10, 15 centimeters, that draw some of this litter down into the soil itself, so increasing the organic material within the soil itself that they feed on. And then there's the deep burrowers that bring material deeper down into the lower part of the soil profile. These can be down to half a meter deep. Potentially some of the larger earthworm types that you see here, and they tend to help mix the soil, but also provide drainage channels for water to percolate through the soil as well. And what do earthworms like? Well, they're very similar to the crops that are growing within the soil in many cases, and so are a good indicator of good conditions for good crop growth. They don't like acidic soils. Anything less than about pH 5 in water will show a dramatic decrease in the number of earthworms that you find. Some are affected by fertilizers and fungicides, so this can be showing that they might be also affecting the crop and the crop growth. They like aerated soils, so soils with good structure that aren't waterlogged. And so again, they're an indicator of problems that may be developing within the soil itself. And so they can be affected by soil structure damage. So again, an indicator if there is compaction within the soil itself. And they also don't like soil that's been too cultivated. So the more you cultivate the soil, the more you disturb the earthworms, the more you disturb where they prefer to live. And so in cereal or arable crops, you tend to find less earthworms than you would in an undisturbed soil underneath a grassland sward. But the casts that they produce can help in topsoil regeneration. They bring soil to the surface, and also this brings nutrients to the surface as well. So they're almost like the biological tractors within the soil itself. And this is some information, some results we've seen from an experiment we did down at the Dairy Research Centre near Dumfries in southwest Scotland, where we looked at the total number of mature earthworms that we found in areas that had been treated either by trampling, driven over by a tractor, or in areas where there'd been no compaction on a grassland sward. As you can see here, there was a definite significant increase in numbers in mature earthworms in the non-compacted area, and where there had been compaction, you saw a reduction in mature earthworm numbers. However, this wasn't the whole story. If we were looking at earthworms in general, the total numbers, and looking at immature earthworms, then there was more in the tractor compacted areas, showing that compaction can affect development of the earthworms. So they're not coming through to full maturity, they're not being given that chance to reproduce, and so potentially affecting the future numbers of the earthworms there. And if we looked at earthworm species in particular, and this is one of the species that seem to be affected more by the compaction, and in the compacted areas, we found less of this earthworm in comparison to the no compacted areas. It's got a complicated Latin name, but there it is, a picture of the earthworm we're talking about, commonly known as the grey earthworm. And again, this is some data from a farm in the northeast of Scotland. This was the earthworm numbers from different blocks of soil that were taken from different areas across the farm. And when the consultant showed me this, they were saying, well, I don't really see any pattern to this at all. There seems to be some earthworms in some areas, but not in others. We can't find any in certain parts of the fields that we tested. So I asked them to tell me whereabouts in the field they were collecting these and which fields they were taking these samples from. And with that information, suddenly everything becomes a lot more clear. In the grassland field on the left-hand side of the graph there was earthworms only found in areas that weren't in the wet areas or weren't near a cattle feeder where there'd be probably more compaction. And again, in a field that had byproduct added, a grassland field, we'd expect more earthworm numbers. You were finding that heavily compacted or heavily used or trafficked areas of the field, then that's where they were taking the samples from, and you weren't finding any earthworms there, indicating a poorer soil quality there, probably to do with the compaction of the soil. In a grass lay that been down for 5 years, you saw this increased number of earthworms showing a better soil structure potentially, but at least better soil quality for growth there. In the cereal stubble, you were finding reduced numbers of earthworms, and again this is probably to do with the perturbation of the soil there, the mixing of the soil, the more regular ploughing, and reducing the earthworm numbers. But again, as you move towards the right hand side of the graph, you're seeing more earthworm numbers in the grass lays, reduced where there's been poaching at the surface, so again the effect of surface trampling there. But when there's been byproducts added, then you start to get more earthworm numbers. And the grass lay that have been down for 5 years plus a digestate where you're adding the food source for the earthworms, this is where you're seeing the maximum number of earthworms. So the earthworms then can tell us a great deal more about the quality of the soil that you're finding in these particular fields. And also, this is just to emphasize this point. This is another piece of work that was looking at the different groups of earthworms that were found within fields, where sewer sludge didn't really seem to affect the percentage or the numbers of earthworm from the different habitats within the soil profile. There were similar numbers whether the waste was sewage sludge was applied or not. But with distillery waste, they seem to have a great deal of an effect on the litter feeders, increasing their numbers and the number of groups present within that field in conjunction with the shallow burrowers that seem to be affected by the application of the earthworm applications. So again, it can show an indication of the kind of food source that's being applied and how this is affecting different earthworm groups within the soil profile. There are other methods that don't need, that are quite visual but don't need earthworms. Some of you might have heard about the underpants method, where you use cotton underpants that are buried in the soil to indicate biological activity. This is more of the fungal and bacteria activity within the soil. And this is a pair of underpants that was buried where they've been over winter ploughing, and it just been ploughed and left over winter. And the breakdown of the material within the underpants wasn't that great. However, when there was a cover crop in an adjacent field, then there was still much more biological activity within that field. So they were turning over the nutrients, they're making that ready for potentially whatever crop was going to be grown in the preceding year. And there was this was indicated by a much greater breakdown of the underpants material itself. And I mentioned earlier these bait laminar sticks. These are sticks that have got little wells with nutrients that the bacterial or the organisms within the soil can feed on. And this can show both biologically active soils, like the one at the top, where there's gaps, there's little, the food sources are missing, they've been all used by the organisms within the soil, but not in some depths, against the non-biologically active soil or less biologically active soil. So indicating that there's something there that needs to be addressed as far as the soil quality is concerned. But also with the biologically active bait sticks, because we know where they are in the depth of the soil, then we can investigate what's happening towards the surface in this situation, why that surface area isn't quite as biologically active. It could indicate that there's compaction there, or a lack of nutrients, or some other constraint that's reducing the overall soil quality. So these are good indicators of what's happening at different depths within the soil. So in summary, the soil is a living component. It's a living entity. There's a lot of activity going on in the soil, or there should be. And this is important for both crop yield and crop quality, ensuring that the nutrients that are added to the soil are made available for the crop to grow. And so these organisms, especially earthworms, can be an indicator of soil health and the activity that's going on within the soils. And earthworms are good because they're identifiable. You can see them. You can use keys to identify which earthworms there are. And these also, particular species, may give even more information about what's happening within the soil. And earthworms respond to soil conditions. They like the similar conditions that we want for good crop growth and crop yield. So if the earthworms are happy, if there's large numbers of earthworms, then this can indicate that there's the soil is of good health, and we can try and maintain or even improve that to help improve crop growth. And just to move on to something that we've been doing at SRUC, looking at management changes to use a kind of visual tool. And this also incorporates research that have been done in general about the effects of changing management on not just the soil pH, but also some of the soil biology as well. And this is an interactive tool. You can put in the type of soil that you've got within the field or on the farm, the climate that's generally found in that particular area, and the kind of cropping system that you're interested in. And then you can put in what you think you want to change the management of that field from, and that can be to no tillage or reduced tillage, to adding higher levels of organic matter or higher levels of N organic matter, using a cover crop, increasing the pH, or improving the drainage. And in this case, we're looking at changing the management to adding more high N organic matter. And this then gives you some indications of what that management change would be. Looking at margins, or in the short term and the long term. But green are the effects that it should have on some of the biology and the chemistry, and also the structure of the soil. In this case, the adding of the organic material is positive for earthworms, especially positive for positive soil biology, pretty nondescript as far as reducing slugs, or maybe a bit of care needed apart from the number of weeds that you might be getting there. But saying that it's positive for adding soil organic matter to the soil, which is always important. Good for P. Nutrient retention, maybe a bit of concern, but it should overall be helpful for yield. And in the box to the right, it tells you a very brief description of what the advantages are, And if you wanted to look to see where the research on this was done, then there's links through to the references that tell you in more detail what the researchers have found as far as adding this kind of material as a management change to your crops. So the rationale behind this was to make something that was relatively complicated or complex with a lot of varied outcomes to something that was easy to visualize and hopefully highlights what the concerns were, but also what the positives were. And to give an overview of what the general responses would be. So you would be able to look or plan in future, if you're going to change to this management, what you would need to be looking for if there were any challenges to do that in a particular soil type, in a particular climate, and with a particular rotation or crop that you were growing. So I hope that's been useful and helps open up a bit of the world of the soil biology. This is an ongoing research area at the moment, and we're finding more and more about this at the moment. Some of the techniques that I've mentioned are relatively expensive at the moment, but hopefully as they become cheaper and more commonplace, then such things as the soil DNA extractions and the communities living within the soil will get more information about that, and these will be able to help to increase our understanding of what soil quality is and how this interacts with good crop yield, resilient crop yield, and maintaining crop yields into the future. Thank you very much. Right. Well, I'm not going to thank myself for that, but hopefully that was useful for people. It's quite a rapidly evolving area of research, and these things are being presented more to farmers as ways of thinking and considering and potentially helping improve soil biology. So an important area, I think, to look at and be aware of, especially for the future. The next presentation will be by Professor Christine Watson. She's the Associate Dean of the North Faculty of SRUC, and her research interests include nutrient cycling, especially nitrogen and phosphorus, but also interested in soil management and worked in a variety of both conventional and organic farming systems, including arable, dairy, outdoor pigs and agroforestry. Christian also holds a guest professorship at SLU, the Swedish University of Agricultural Sciences, in the Department of Crop Production Ecology, and also does teaching on postgraduate courses in organic farming. I think Christine is going to look at some overview of soil quality now for the last part of these presentations.
Hello everyone. My name's Christine Watson and I'm from the Department of Rural Land Use in SRUC, and I'm here this morning to talk to you about taking soil health one step further. I want to build on what you've already heard from Joanna Cloy and from Paul Hargreaves and think about how we integrate that information and how we link soil health with plant health and human health. We've heard quite a lot today already about possible indicators of soil health, the biological indicators, the chemical indicators, and also the physical indicators. And here you can see a group of farmers working together as part of the Soil Biology and Soil Health Partnership funded by AHDB and BBRO. And here they are in a field doing some visual soil quality assessment and discussing the findings and discussing how they might improve the soil in this particular field. What I want to do is to get you to think about how the chemistry, the physics and the biology all link together. The idea that we need to think about managing all of these parameters within soils when we start to talk about soil health. I think we're all very familiar with the idea of habitat creation when it comes to landscapes, when it comes to a farm or a field or a hedgerow, we're familiar with the idea of planting different crops or non-crop species to create habitats for beneficial insects, for example. We're all very familiar with the idea that certain plants grow under certain conditions. So here we can see bog asphodel and the idea of the habitat in which that will survive. I think it's rather more difficult to think about the landscape below ground, and I want to build an image in your mind of the idea of a landscape below ground, of creating a habitat, and what it is that we do as we cultivate the land and manage the land in different ways to create a habitat that will give us the beneficial soil biology that we want to produce strong and healthy crops. Many of you will be familiar with the Woodlands Field experiments at SRUC at Craveston, where we have a long-term experiment on soil pH which was established in 1961. It's an 8-course rotation with soils managed from pH 4.5 through to pH 7.5. And if you look at what happens at pH 4.5, you see that the oat crop creates a much better yield than either barley or wheat. All 3 crops are quite happy between about 5.5 and 6, but once you get to pH 7.5, the yield of oats is very small and the barley does slightly better. We're interested in understanding what it is that's going on below ground, what's going on with the biology, the physics and the health and the chemistry that actually create the conditions for good crop production. And here you can see the soil health scorecard, which many of you will have used, carried out for the pH plots. And you see in the diagram on the right with the traffic light system of green is good, yellow is something you need to think about, and at red you need to investigate and possibly take action. And we see, for example, that at pH 4.5, very low numbers of earthworms, and we also see that some of the micronutrients start to get locked up, and we see here, for example, magnesium not being so available at pH 4.5 as it would be at pH 6. The next stage is to try and understand what soil biology is there and think about what it is that soil biology is doing, and we're investigating with colleagues, particularly in the University of Lincoln, some molecular tools for understanding the soil biology. I want to show you a published example of where we have people using molecular techniques to understand fungal community structures in soils. This is an example from the Swedish University of Agricultural Sciences, where they have looked at fungal community structures underneath 4 arable crops: oat, wheat, oilseed rape and pea. You don't need to understand enormous amounts about the detail of what they actually did, but you can see that they have looked at soils with inversion tillage and non-inversion tillage, and the big separation in the diagram on the right between the fungal community structures in the soils under the 4 different crops where there has been non-inversion tillage in the grey, marked in grey, and compared with soils where inversion tillage has been taken place, marked in white. There's a lot more to investigate in this field, but it's quite exciting developments that are taking place in terms of thinking about how we manage soils going forward to the future. We're also really interested in understanding the links between soil health and plant health, and how we can use the kind of simple soil indicators we've been talking about to help us understand and manage plant health. And that's a question we will be investigating in the new AHDB strategic farm in Scotland. We're also interested in how we can help people to understand the importance of soils in a wider context. And this diagram, this table, comes from a paper we wrote a couple of years ago, where we were looking at the types of connections between soil and people. And the first example, which is a direct connection, might be teaching land managers to use visual soil evaluation or to describe their soils in order to understand soil quality and therefore to manage it better. But it's also really important to think about the links between soil and food production through food quality and into human health. And that's sometimes quite hard to get people to think that soil is important in terms of determining the quality of the food that they eat. So we're interested there in working with the wider public, so gardeners, cooks, school children. And then it's also, I think, quite important going forward in the light of climate change that we understand relationships with soil degradation and how important it is that we understand how degraded soil can reduce food security and what we're able to do about it. So looking towards the adoption of a more sustainable farming system. And that might be something that we think about in terms of policymakers. To do all these things requires improved communication about soils. And on World Soil Day on the 5th of December, which as many of you know takes place on the 5th of December every year all over the world, as it's the United Nations World Soil Day, we were involved in launching a new platform called UK Soils, which brings together information to inspire us all to learn more about soil and to help build communities to take actions to improve soil health. Just to give you an idea of what the website looks like, here it is. And here you can see where anybody can take part in a public survey about perceptions and priorities for soil health. But there's also a building amount of information. It's early days yet for this particular platform, but the idea is that there will be information here which is open to everyone and which will be applicable to many different audiences. So we have soil on the farm, but we also have information about where you can study soil and about soil in culture and history and the arts, and also soils in medicine. The idea of UK Soils is that it will also help to advertise soil health pioneers. So if you have a lighthouse farm or a living lab or a kind of soil assessment network, then here is the opportunity to publicise it. And as you can see, Scotland's looking a bit bare at the moment, so we need to fill in some of the excellent work that's going on in Scotland. So the UK Soils Network is a fantastic resource to get involved in. Leonardo da Vinci said we know more about the movement of celestial bodies than about the soil underfoot. Science has moved a long way since Leonardo da Vinci, but there's still a lot to understand. And thinking about how we use our knowledge of soil biology, soil physics and soil chemistry to create habitats below ground that will allow soils to deliver for us in terms of crop production is certainly an important way forward. Thank you very much.
Well, thank you very much for that presentation, Christine. It's definitely some food for thought there, but also some interesting sites to go and visit as well to be hopefully more proactive in the soils community and understanding and promoting soils and soils quality, which I think is important to us all, really, especially in the farming community, where this is really something that you need to consider and try and maintain as healthily as possible. So we've got a bit of time now just to go to some of the questions that have been posed during the presentations there. I don't know if Christine is around and wants to come in on this and see if she wants to answer some of the questions as well. Thank you, Christine. That's brilliant. I mean, the first thing that came in wasn't a question, but more of a comment. It was saying that we need to get Scottish First Ministers to take soils health seriously, about 3 years behind our English counterparts. Which I mean, I'm always an advocate of promoting soils, and I have done that at Holyrood in the past. But yes, I mean, I can only agree with that statement, and I don't know if you've got anything else to add to that, Christine.
No, I mean, I think we are obviously very keen to support that idea, and willing to work with people to find novel ways of bringing soils to a greater public attention and therefore ministerial attention.
The next one's a very specific one about soil chemistry, saying about scientific evidence that balancing calcium and magnesium ratios have a positive effect on crops. I mean, I'm aware that there is some research looking into crops and the calcium magnesium balance as far as crop growth is concerned, and the importance of having sufficient levels of both of them, but some of the ratios also tend to put too much emphasis on the ratio itself and not just ensuring that there's sufficient amounts of both of them. I don't know if you've got anything else to add to that.
No, I think sometimes we have to be very careful that what we're reading is applicable to the soil types that we're working in, and ensuring that that we think through where you are, what the soil type is, and therefore what is appropriate.
The next one's quite a specific one and quite interesting. I've not really thought about this before, about selenium, which is obviously a micronutrient, important, but it is important in both animal and plant health. And is there an organic method to fix this, or what else would you do? I'm not aware myself of organic material having high levels of selenium itself, but I just thought in the past it were related to the soil type and the underlying rocks, how much selenium you had, and potentially if it was deficit, then I would be suggesting maybe within the mineral licks or whatever themselves. But I don't know if you've got any more experience of that.
No, although in the team in SRUC we now have Dr. Jacqueline Stroud, who is an expert on this. Yeah, selenium, if there is a deficiency in the underlying geology, you can't manufacture selenium in the soils. And obviously mineral licks are a possibility, so are fertilizers. But my understanding is there aren't available selenium-only fertilizers. You can get compound fertilizers that include selenium. But clearly it's a really important one. I think it's quite topical at the moment because selenium has potentially been shown to link to our ability to fight viruses.
Yeah, no, I'd agree there as well. There's a technical one for shear strength measurements. Do you need to remove the turf first? I would say that probably not, because you're looking at the top part of the soil and the integrity of that with your shear strength. So the rooting mat and the turf is important, as long as you can get your the blades into the soil. Next one. App recommendations, always interesting ones, and it depends which where you go for. I mean, there's a multitude of these now, from Potash Development Association through to a couple from SRUC as well. I think it's just been sensible that they come from a reputable site, and that they, understanding what information they're drawing on really to get your decisions from the data that you're being asked to put in. I don't know if you've got any particular favourite apps that you prefer.
There are so many out there now, and a number that have been developed in, for example, EU project. There are a lot that claim to do very similar things around visual soil quality assessment. But I think you're right, Paul. I think, look at where it came from and, who developed it in terms of the reliability of the information. I don't think I could actually recommend one. Obviously, I see one.
Is the time of year important for VESS and worm counts? Well, that definitely is. Having done numerous VESS and worm counts, then trying to do them or demonstrate them in the middle of the summer is usually a fruitless task, as you struggle to get into the soil and find any worms because it's too dry. So generally spring or autumn is the times when we recommend you take these samples or do these assessments. A, because it's easier to get into the soil, and B, that's where the activity's starting, or it's the soil starting to wet up again. in the autumn time, where you should get this the activity, especially from the worms, are more prominent as well.
I think the important thing is that if one of the great things about soil health and doing these kind of soil measures is looking at what happens in a particular field or after following a particular management practice or a particular crop. And I think the important thing is just to go back at the same time year on year, so that conditions are never the same year on year. We all know that. But I think if they're to be in any way comparable and therefore help you, then to actually try and go for a window in September or May might be a good way of doing it.
Yeah, no, and I think it can also be dependent on the problem that you're trying to identify, and so giving you time to rectify that if need be through the year. So sometimes spring is better for that, but sometimes autumn if it's something that you're going to do in the next part of the rotation. The next one is, what is the VESS app? This is I think an app that was developed by people in Switzerland, where you add information to drop-down menus on your phone or tablet in the field as you pull apart the blocks of soil, and then this helps you assess or give a score for your VESS soil structure as you see it in the field, and from there hopefully you can then make some management decisions depending on the severity of the effect of on the soil structure that you've seen. I think there should be a link through to that from the SRUC website, linking it through to, if you type in VESS in there, it will take you through to a web page that's dedicated to VESS, explaining VESS and the different kind of research that's come out from the development of that.